Good morning, everyone. Thank you for joining this other webinar for the grant scheme activities of ECPGR. This is the second webinar that we are offering this year. There was another one on Vitis sylvestris earlier on. All these webinars are recorded and can be watched again from the ECPGR website. Today, we introduce an activity that was submitted and carried out by two working groups, the on-farm management and conservation and the wheat working group. And this is focused on a very special crop that is called Binkel in Germany, club wheat in English. It is Triticum aestivum subspecies compactum. This is an underutilized crop that grows well in marginal mountainous areas. And the project was joining especially alpine area countries where this crop can be grown. So from Germany, Austria, Italy, France. And the idea was to preserve and to check the diversity of this existing crop and also promote its utilization. So it's a very comprehensive type of project. The project was submitted by, as I said, the two working groups, and specifically Albrecht, Serfling, the chair of the wheat working group from Julius Kühn Institute, and Rudolf Vögel from the VERN Association for Conservation and Rekultivation of Crops. So now the two guys will explain what they have done in a period of 3 years, from July 22 to March 24, reaching a lot of results, interesting results, I would say, with little money, because these projects, as you probably know, allow the use of between 30 and 60,000 €. In this case, it was 37,000, so a small amount of money for a lot of work. So I already congratulate for the success of the two and the teams. Having said this, I would pass the word immediately to Rudolf Vogel, and the presentation will be shown on the screen now. The floor is yours, Rudolf.
Thank you very much, dear Lorenzo. I'm very happy to find you again. We missed a lot of couple of years now, I think, but we have been in very narrow contact on our project. Triticum compactum, thanks for your introduction. I think Triticum compactum is listed as an own species of the genus Triticum, but it's nearly extincted, it's forgotten, it's out of order. It's the same level as Spelta, or also hexaploid, of course. And we thought with the project it might be a very interesting work to have a better and narrow look to this thing. It is in Europe, it's forgotten, it's out of use since nearly 1 century now, but there is also an older breeding history in Europe, and it's an actual use in the United States of club wheat. Please give me the next slide. Yeah. The presentation is divided up. For my part, I will give you some information about the background of the project, the collaboration of our group, and the informations about the club wheat accessions documented in gene bank collections, information about our field trials and the ongoing work. Afterwards, Albrecht Serfling will give you very good information about his work on the pathogen resistance and the genetics of the selected clubweed samples. Next picture. Yeah, the species Triticum compactum, it's neglected, it's underutilized now, it's nearly extincted in use. You see in the picture the very outstanding performance of the clubweed ears. It's an interesting thing we found out. It's also out of the seed policies and out of breeding in Europe, actually, but it has a historical background. Next slide, please. We know that crops are moving. The moving of clubweed, it's very outstanding, fantastic. It's given clear that clubweed was brought in the after the medieval age. It was brought to South America, and it was brought also to Mexico, to Colombia, and it was moving afterwards to the northwest of the United States, maybe Canada also. And we have a lot of documentation of clubweed in the native local European regions. Next slide, please. So there are historical sources from Russia, from Siberia, from the late 18th century. There are also sources from Scandinavia, from France, from other parts of Germany. Also Neolithical sources from the very early period in Europe, 4,000 years ago. Here you can see right down the actual ears of clubweed from the breeding efforts in United States from the 1930s of last century. Next slide, please. And in Europe, you find some local names of Triticum compactum. In Germany, as Lorenzo give, Binkelweizen, Igelweizen. In France, it's named as Harrison. In Portugal, Spain, it's called Mochon. In Scandinavia, it's given as Kupweisen. In the Slavian, Russian, Ukrainian, and more eastern part, it's called Teremok. And in the English speaking world, Great Britain, United States, but also Australia, it's named as clubweed. We brought up EURISCO research, data research of the Triticum compactum accessions in our gene banks. We found out 1,300 samples there, and when we selected for the origin, we looked for European origin for a more safe, more approved taxonomy, and maybe a little part of description data. We brought up 241 selected accessions in different countries. You can see the Ask Gene Banks in the circle below. Next slide, please. What was our project work? We had to do baking tests, experiments, quality analysis of the samples we are able to bring up in field experiments. We had disposals to interested farmers, bakers, groups. We had some seed multiplying, transformation, and the start of the establishing of a new on-farm conservation with interested farmers. Next slide, please. In this work, we had also a very fine collaboration with the Slowfoot group. Here you can see our field survey plots. We had 3 years to spring accessions in 2022, and in 2023 we tested winter accessions and spring accessions in different sites in Tyrol, Italy, Austria, in Bavaria, the LFL, and at the VERN site. And in 2003, we tested also with our French colleague, Clément Debiton from INRA in Clermont-Ferrand. Unfortunately, there were also only description data available to get because of lodging, very bad weather. The field experiments failed. The parameters we listed was yield, agronomic data, tolerance, resistance by field survey, and the cultivation value in general, also a phenotype description with UPOF data. Next slide, please. Here you can see some impressions of the clubweed field experiments. Left up, there is a modern standard of spring wheat, and all others are Binkel wheat. You see, it's not so high in growing, and you see the different types of ears with spikes, without spikes, and you can see also the problems right down with lodging of some samples, and you can see the problems of pathogens, the Kärntner Winterbinkel, an accession we get from an Austrian breeder with hard performance of yellow rust, stripe rust. And left down you see our North German field sites of VERN collaborating with our local university. Next slide, please. Quality of trial harvest here. We were surprised about very good performance of the protein content, mostly of the spring wheat accessions. You can see a very high level of sometimes more than 20% of protein content in the dry mass. It's very good also for a spring wheat. And also in the winter accessions, you can see a level of sometimes more than 50% compared to the modern standard, very wide right, the standard Tobias. It's much more better. Of course, this means connected with the agronomics that the yield is not so high. Next slide, please. You can see left as an example, the yields of spring club wheat, the relative yield of all field sites we had in 2002 and 2003. It's changing from 140% relative down to lower than 60%. It's very varying, of course, differing from the soil quality, from the precipitation, from the site conditions in general. And you can see in the right slide, the right graphics, that the standard spring wheat Tobias brings up in our in our North German field site about 6 tons per site per hectare, and our tested accessions are much more lower, so we have in general a reduced yield of 1/3. This means it's guilty for our German site, and it's not guilty for some less favored field plots in Italy, in the Tyrol Alps, of course. Next slide, please. We had a very big work on testing the baking quality of club wheat. In general, we presume that clubweed in alpine area was not so much used for baking in the history. It mainly was used for soup, for cooking, not for bread making. It's interesting that for the homestead test we had, for the baking quality tests from LFL in a baking laboratory, that all tested samples were quite good and quite acceptable for making bread in a homestead, in a homestead way in general. Our colleague in France, Clement, offered us also a modern industrial analysis with an alveograph called Chopin test. And with this Chopin test, we were able to different most of the tested clubweed samples as not very suitable for a modern industrial use. Maybe 2 or 3 tested samples were useful, but most of them they didn't were able to use the alveograph test successfully. Next slide, please. And by this, I will give to Albrecht Serfling. In the end of the presentation, I will give you also some informations of our on-farm groups, of our farmers in the Alpine area interested and in charge now for the best samples for on-farm use further on. Please, Albrecht.
Yes, thank you for this introduction of the clubweed, Rudolf, and Lorenzo for the introduction of me. And one part of this project was how we can differentiate the Binkel from the common wheat which we have. You have now seen the spike numbers increase, the ears are shorter, and that's the reason why this Binkel is called Triticum compactum. And here you can see a field picture. And if you go to the next slide, Noah, we can see the difference. And the main question of my part of the project was are there genetic differences between Binkel and common wheat? And can we see, using marker or SNP analysis, what is the difference between the normal wheat and Binkel? And Rudolf mentioned already that many of the cultivars or accessions which we have from the Binkel under cultivation are susceptible to the rusts. And in order to improve the yield and the quality in the future, it would be a good advantage or a challenge to introduce resistances or resistant accessions into the gene pool there, which is used for cultivation in the future. Yeah, next slide, please. Yeah, and before we started the project, we got preliminary results with a few of these genotypes, and you can see here these 5 accessions or cultivars which we got here and which were investigated. And that was the first trial to differentiate genetically between Binkel accessions and different wheat accessions from gene bank material. And at the moment, it is the case that in gene banks you have sometimes a mix of Triticum compactum accessions. Some are shown there as a normal wheat or normal wheat accessions, but these are compactum accessions, so that the data from gene banks are sometimes not reliable. Therefore, a marker for the differentiation would be a big advantage in order to generate Binkel cultivars in the future. And how we can do this that I will show you now in next slides. You can go to the next slide. And these preliminary results showed here that the Binkel accessions are, at least these 5 Binkel accessions are quite different from the typical winter wheat cultivars which we find in Germany here, and from other wheat cultivars. These data are not published, and the problem here is in this study that the winter wheat cultivars in Germany show us not the entire gene pool of the wheat, so it's they are very close related because they are selected for yield and quality, and therefore there is not such a big differentiation possible as you can see here regarding the land races. So next slide. Yeah, and what we have done is then that we got 227 Binkel accessions from different sources. This were the gene bank of the IPK in Gattersleben, but also the Kühn from the Department of Agriculture in the US, and some other gene banks which are involved in the European evaluation program. And we have nowadays the possibility to use the reference genome of wheat so that we can order all the SNP markers which we get in such analysis there. And we use for our analysis here the so-called 25K chip that allows us to use more than 20,000 markers which we can use for the differentiation, and which we can also use to map SNP markers there, and which are usable also for the identification of Binkel-specific introgressions, maybe SNPs, which we find only in the phenotype of Triticum compactum accessions there. And so that makes possible that we can compare SNPs between typical wheat cultivars and Triticum compactum. And furthermore, we can then use this region, which we could identify by the higher number of SNPs in comparison to the genome in the background, that we can blast this to publicly available databases, and that we can develop so-called cusp markers. These are competitive allele-specific PCR markers, which allow a very simple PCR reaction where we can select the Triticum compactum genetically. Yeah. Next slide, please. Yeah. And one of the first results was then that we could, based on these SNP analyses, see what is the background or the regional background of the origins. And what we can see here is that you see that compactum lines, if they come from a specific region, are clustering. And you can see here, in example, in case of the US and Poland, from which region the original lines derived, and which of these lines were used for breeding activities in the countries. It makes it possible to separate different regions. You can see here the lines from Kazakhstan, Kyrgyzstan, from Asia, this Termok lines, they are closely related. And so independent from the data shown in the gene banks, we can see what is the background or the regional background of these Binkel accessions, and can use this for further investigations and for breeding. Yeah. Okay. Next slide, please. And what we can do also is, based on the SNP data which we have already from the wheat, we can compare the genetic identity with the wheat cultivars which are grown in Europe at this time. And you can see that the wheat cultivars are close related, shown here by the arrows and this orange dots there. And you can also see that some of the Binkel accessions are not related to these wheat cultivars. They are far away, especially if they are from regions where they are cultivated or grown, which are geographically far away from Europe. But you can also see that some of the Binkel accessions are within this gene pool, so that a simple comparison of the entire genome and the SNP data is not enough to define a Binkel accession genetically. And therefore, we try to analyze what is the genetic background of this compactum phenotype. You can go to the next slide. Yeah, that is here. Yes, thank you. And what we have done there is that we compared all the significant Binkel or clubweed phenotypes to the wheat phenotypes. And by the simple comparison of SNPs, which have been mapped using the reference genome, you can see that you find in specific regions in the background. You can see here the chromosomes and the example of chromosome 5a, an increased number of SNPs. And this is a sign for an introgression in the background, which comes actually from the original Binkel lines, or which is linked to this compactum phenotype. And using this information in the background, you can select physical regions in the genome, which you can blast and where you can identify candidate genes. Yeah, next slide, please. And the entire information by the SNP markers you can use for a genome-wise association study. That means all of the wheat and compactum genetic information is analyzed together, and specific markers or specific markers in a region which are associated with the compactum phenotype, you can see here as a tip or a higher value of this so-called lot value, which is the negative logarithm of the p-value by statistical tests. And this lot value of 6.5 shows you that you have more than 100,000 times higher possibility that this marker is linked to the compactum phenotype than the noisy background. The threshold was 5.74, and these regions are already known for a compact ear, but not directly for the compactum phenotype. And one of these QTL, which you could identify here, is this QTL on 5A. And in the literature, you'll find actually directly the evidence that this background information is related to the compactum phenotype. You can go to the next slide. And using the one back, please. Using this marker on 5A, you can, or we developed CUSP markers, this PCR markers, where one primer shows the SNP and one other primer not, and they are marked with a staining, so that we can differentiate here using this region successfully between the compactum phenotype and the wheat phenotypes. And you see here the phenotypic results. All genotypes which show the SNP here as a orange column can be differentiated and show us we have really a compactum phenotype, and this is really well associated with this compactum phenotype and can be used in the future to identify an example from gene bank material very easy what is a compactum genotype, and can use it maybe for future breeding. Yes, now we can go to the next slide. And we looked in literature on chromosome 5A, we find this typical information about the Q locus, which is related or associated with a higher number of spikes with a shorter ear. And in other sources, we can see here also the QTL on 2D, which we could identify. And if we find these two QTL in the background of our investigated material, we can be sure that we have a Triticum compactum phenotype. And this will easify and accelerate the process of identification of compactum lines, because we have not to wait for the ear development, and we have not to count and measure the spikes and so on. It's a question of a few hours that we get these PCR results. Okay, you can go to the next slide. And another very important task is the identification of resistances against leaf rust. You can see here the typical symptoms. Rudolf showed already what you can see in a field trial if you have a susceptible cultivar. Here we can see the typical symptoms, this rust pustules. And we could see in our first results that many of the genotypes are more or less susceptible to the leaf rust. In this case, I will focus more on leaf rust now, and only a few or a part of the identified genotypes there, or compactum genotypes, show resistance against the leaf rust. However, the genotypic effect is significant. We have a significant effect of on the phenotypic variance here. And so we can use this phenotypic information together with our marker information to identify loci which are associated with leaf rust resistance. You can go to the next slide there. And so you can see here it's quite different from the identification of genetic background of compactum phenotype. We have here low lot values, because if we have this resistant genotypes, there are many genes in the background expected. And what we could identify here, that's the information framed in red on chromosome 5B. This could be a new resistance resource in the background, which we could identify in the cultivars of Gelber Igel or Gelber Igel accessions, an example. And using this information, it makes now possible to start the breeding with regard to more or better resistance to leaf rust, which is actually the most important rust. And so it is linked also to quantitative stripe rust resistance, but that we have to check in more field trials in the future. But one of the candidates will be one of the Gelber Igel accessions, which can be used then for the resistance breeding in the future. Okay, we can go to the next slide. Yeah, and yeah, okay, thank you. I will come to the summary. We could differentiate phenotypically and much faster genotypically the Triticum compactum accessions from common wheat cultivars using these molecular markers derived from the SNP analysis there. We could identify the origins and relations of Triticum compactum accessions by principal component analysis. We could identify leaf rust resistant compactum accessions which are usable for breeding in the future. And furthermore, we could identify QTL on chromosome 5B, which is linked to a possible unknown resistance to leaf rust. And so the genotypic data of many compactum gene bank accessions are now available and usable for genome-wide association studies after phenotyping, but also, and that mentioned Rudolf very well, other traits, in example, quality of products, yield, and many other traits. And now I want to thank you for your attention. So that's the next slide, I think. And the partners which are involved in this investigation, and the partners also here in the Alpine region which performed the field trials, and you for your attention. Thank you very much.
Yeah, okay. I will summarize a little bit. Thanks, Albrecht, for your very keen and professional explanation. It would have been impossible for me. It's not my work, of course. You see in the background a typical alpine landscape, and combined with a club wheat field trial in the middle of the Austrian Alps near Innsbruck. It's the site of Christian Partl. And for this, I will have to give my thanks for the project collaboration, special to Clement de Beton to INRA, Christian Partl in Innsbruck, Manuel Pramsola in Limburg, Italy, Herbert Steiner of Slow Food, and the Bavarian colleagues Klaus Fleissner, Ulla Konradl, which had some special work on club wheat before, and all integrated and involved farmers and bakers and external colleagues, which get information about our work. On the right down, you see one of our typical Italian farmers from South Tyrol in a level of 1,500 kilometers. He started off recultivation with a sample of club wheat we gave to him. And actually we have about 4 active farmer groups in South Tyrol in Italy, in the Austrian part of Tyrol, and in Bavaria, and also in the northern part of Austria in the Salzburg region. I hope this project may give a special impact to our ECPGR groups, combining the scientific work with farmers' work and with the possibility of recultivation and re-establishing extincted, forgotten crops. Thank you.
Thank you very much. Thank you very much, Albrecht and Rudolf, for this very interesting presentation. Very well done. It is now possible for our audience to ask questions. You can either write in the Q&A, or you can ask for raise your hand, and we can let you ask a question directly. Let's see if anybody has a question. In the meantime, while people think, I can ask Rudolf, what is the perceived impact that this project gives to the possibility to improve or expand the cultivation of this crop? Could you say something in general of the impact of the project, or your perception?
Within the time of the project, we got a lot of questions from people and groups interested for marketing club wheat, not so much from farmers, but also for farmers. So I think the impact for reintroducing club wheat is mainly concentrated in mountain areas, in the alpine parts of France, Swiss, Austria, Italy, and also there is an interesting response from mountain farmers. Small sites, and the agronomy of mountain farming, mountain cropping, it's not so high. It's a kind of mixture, of course, but it could be a kind mixture combined with tourism, with history, with culture, and nevertheless in some mountain areas in Italy and in Austria, you have also smaller processing factories, smaller mills, smaller bakers, and not so much industrial performance on crops. And this might be a chance to stabilize this kind of neglected, underutilized crops. But it's a hope. It's a try.
Yeah. Thank you, Rudolf. Okay, we have a question from Nora Castaneda. What is the content of gluten in club wheat compared to spring winter wheat? Do you have an answer?
It's to me.
Whoever can answer.
um,
If you can.
when you realize gluten as a part of protein content, it's also higher than in spring wheat. In general, it's higher than in winter wheat. But we had no special analysis on the problems of gluten wheat for dietetics. If the question is in this direction, we had only analysis for the quality of protein and gluten for a baking process.
Thank you, Rudolf. If you want to raise any hand, you can use the React button on the bottom of your screen, and there are the hands that you can raise, or you can still formulate a question on the Q&A. I have another question in the meantime. What is the availability of the accessions that you have analyzed? Can they be requested to gene? Are they deposited in a gene bank? Can they be requested? Has the conservation of this material ex situ been improved? Anything that you can say about this?
It's to me?
To you, Rudolf, yes.
Yeah, okay. We have the force from ECPGR, from you, to collect and to store all samples we used, we tested. And for this, our VERN has an own wheat and own a crop collection of more than 2,000 accessions. And the club wheat, we had the field trials. They are now also part of our collection, and they are available. Nevertheless, we are also in charge for the farmer groups. The farmer need bigger samples, bigger lots of the accessions, of course. And for this, with Herbert Steiner, I will go tomorrow or the day after tomorrow on our tour to the Alpine region. We will bring a bigger lot to as a request to Italian farmers, and we try also to serve the other groups for this. But in general, we would like that all used accessions, all proved accessions, will be hosted and kept alive from the farmers by themselves.
Okay, thank you, Rudolf. I do not see other questions. Yes, there is one from Sandra Gorichnik. Rudolf, you showed in the beginning that several gene banks have club wheat samples in their collections. How many of these provided samples for the analysis? So how many gene banks have provided samples for this exercise that you made?
We used samples from the Swiss gene bank, from the French collections, from the German EPK collection, and also from the Austrian very traditional Tyrolean gene bank, which was responsible for the collecting of the last living club wheat parts in Austria in the 1950s. In general, it's very difficult to use gene bank samples for such experiments for field trials because the lots you can get from the gene banks, they are very, very small, as you know, and you need maybe 2 years for multiplying. And this was not our time.
Sure.
Yeah, maybe I can add, we had, Rudolf, we had also from the screen from the USDA some samples, I guess. And we had then, what we have seen is that the material, at least at the beginning, is not always clean. So you can analyze it genetically, and you can see that in some cases, so they have the same designation and same accession number, but you see then that they are sometimes contaminated with normal wheat or so, because gene banks, maybe they are in the field and they are lodging and so on, and there's sometimes a mixture possible. And I think a few of the genotypes which we could select were also available from the European evaluation program, which was managed by Sandra already. A few of the genotypes are also clubweed, but I think it was not always known before. So that's.
So I have to correct a little bit. We used also one sample from an Austrian breeder. It is the only sample, as we got known, which is an original clubweed under the response of the breeding company. It's not offered, but it's in the collection. It is available. Also, we used in the field trials 2 samples from the US, from the commercial clubweed production there. Yeah, that's all.
And by curiosity, what is the origin of the accession that resulted to have a resistance to leaf rust?
Yeah, that was Gelber Igel. Rudolf, you can correct me, but that's German accession. So, or from Germany. And
is that at VERN or in Gattersleben?
No, that's that you can order also from IPK Gene Bank, but Gelber Igel is also available from Austrian or from Switzerland gene banks, and mostly it's the same, but in some cases there are some differences genetically. So it's simply the name which is the same. And I think origin is Germany, Alpine region at least, but origin is Germany. Or is it right, Rudolf? Okay, you are not sure. So,
Problem of description data from gene bank accessions in general. It's the name is German eagle in English translation hedgehog because of the spikes, and there was no information anywhere about it. Nevertheless, Gelber Igel was used in some projects in Germany, and we have it in general in field multiplying. We had also a little bit of selection and breeding and cleaning in your sense, because we found out that clubweed in general in the field production in the history, it was not a single cultivar. It was mainly a kind of mixture of different types. This is according of the agronomic cultivation of the farmers use maybe.
Good. Thank you, Rudolf and Albrecht. Seems we have satisfied the various questions, and it was, I think, a very interesting presentation. So, as I said, this will be available soon from the ECPGR website, together with the other presentations. And I would like to thank all the participants for your attendance. And stay tuned. We will have more of these webinars on other ECPGR activities. And also, there will be a new call for activities coming out in October for the working groups that are interested to submit new proposals. The call will be out in October and with a deadline, I think 1st of February. So you can use the Christmas vacations to prepare submission. Okay, if there is no other comment, I will call this this webinar and wish you a nice lunch and day.
Okay. Yeah, thank you for the invitation and opportunity to present. And yeah, I think we will participate in this call there. Yeah.
Good, great. Goodbye, everyone. Hi, Rudolf. Nice to see you again after many years.
Goodbye.